All right. Hi, everybody. So, if you look at the schedule, you will see that this is a lecture, actually, not a workshop. So we actually won't be needing, like, if you want to take notes, sure, go for it. But this is going to be more of a lecture/discussion. And I know that, it's a third day of the school, and you were working very hard 2 last nights on, let's say, building new international opportunities to international future collaboration on projects, or how we say NGS party. So I know that, and you may be already tired. So I went ahead and get some initiatives for you to actually talk to me. So if you're going to actively participate in this, and you're going to tell me about your experience and your ideas, then you're going to have an opportunity to try some classic Polish candy. All right. So you already know who I am, and I'm going to be running later on during the group projects. I'm going to be running a more specific hands-on hackathon. But this lecture is more about general, culmination of our unsupervised learning plot, and more about the applications of this method in practice. So you already saw some of the way that we actually classify methods for unsupervised learning. Specifically, Kasia and Kaspar did a great job, showing you some examples of how lower rank dimensionality reduction, for example, PCA, can work, and some of the clustering methods. So what I want to highlight here is that in our supervised learning, we don't have labels, and it's based on the exploration. So what we are trying to do is we want to look at the patterns that are hidden patterns that are available in our data. So it's very much fishing, right, for the data. However, the actual applications are really there's so many actual applications, and usually even if you do supervised learning, you would probably do some of the unsupervised learning before to try to take a look at your data, or for example, correct the different batch issues. So we know already something about clustering and lower representation, where we just basically look at the projection of our data in some different dimensions, usually lower representation. So it's easier for visualization, and it's easier for us to notice patterns that can decrease the dimensionality of our data. And you also heard about clustering defining the groups. Actually, there is also a third kind of class of unsupervised learning. Yes, it depends on who you ask. And this is probability density estimation. And mostly it's connected to trying to figure out the actual distribution that your data is coming from. So you get the real data, and you're trying to find a distribution that is as close to actual distribution as possible. And this actually is not as frequently, you wouldn't see this very frequently in your papers, but it's very important if you're doing some method development, because this can be used, for example, to create simulated data, right? So to get actual data and try to create a good representation with some ground truth, because in biology we don't know, we don't have labels. So we just want to get some data set that we can test our method on. But it's also sometimes used, for example, for outlier detection, right? So we fit our distribution, and then we try to look at points that do not really fit, right? And the special version of this is kernel density estimation, and I will show you an actual real-life example later on. And the good thing is that it works for any distribution of your data, but it actually needs a lot of memory, because you need to store all of your data to actually find the good estimation of it. And the main point that Kaspar already mentioned is that the biological data sets are complex, I would really love it if we are still at this point when we can just visualize just two genes and straightforwardly understand what is happening, but more frequently we have situations like this. And this may be actually a good point to ask the first question. Do you know why in like omic data, in genomics specifically, why samples are usually in columns and features are in rows? Anybody has an idea why? No, this is like it's in machine learning. Usually you would see something completely different, but in biology you present it like this. And specifically, a hint may be that at some point, if you would have it completely the opposite way, the number of genes would get constrained to a smaller number, specifically 16,000, around 16,400. Excellent, exactly. Yes, you get a candy. There you go. Alright, so yes, exactly. For a long time there was a problem because this is the maximum amount of columns in Excel spreadsheet. So the biologists would open the data, they would drop some, and then they would pass it on to a person that analyzes it, and that would create a bunch of issues. So this is why we actually do it different way. Right. There are actual papers that were published this way. Well, now we know better. And yes, and also we have to think about the way like multi-omic data, which is what I'm going to be talking about more later on. It's actually like different types of omics. For example, DNA methylation, gene expression, or like chromatin accessibility. They show different snapshots of how of cellular activity within an organism, right? So integrating something like this have a potential to give us the insights into etiology of a disease and how organism actually works. However, it's obviously not trivial. And frequently we also have different time points, and we have time scale data, and this further complicates what we are actually looking at. This here is microbiological relative abundance of species at some sample over the time. So please have it in mind that that's all is unsupervised learning, and because our data is so complicated, we usually don't use just one of it. We're going to see all of the examples actually use more than one also. So the most basic practical application that comes to my mind is batch effect correction. And batch effect is non-biological variation. That's technical variation in your data that's not connected to what you're actually studying, unless you're studying batch effect, which maybe it happens. So I said technical, right? Where you're all familiar probably with batch effect, unless if you are not familiar with batch effect, I envy you. But where is the batch effect coming from? Can somebody give me an example of what could cause a batch effect? I know that some of the people are studying batch effects explicitly.
Different runs of different analysis.
Different what?
Runs. One machine runs.
Oh, runs. Yeah, exactly. So running on different machines. Yes, correct. There you go. Another example. Yeah.
The machine starts to clog up during acquisition of the data.
Doesn't.
Well, it's not completely plugged up, but the intensity changes over time because
so decay, right?
Yeah,
you don't have a decay. True, it's more of a decay is a technical variation. I will give you that. Probably you would see it over the time, so it wouldn't actually use cause a batch effect unless your different samples have different rates of decay. But it's also because of the technical variation. So I don't know if I'm good enough to actually give decay.
Oh, wow.
Different clinical data for different countries, so
So if you're using data from public sources and the data was prepared in different countries and different facilities even, right? It doesn't have to be different countries.
Yeah.
Still, it's a cause of maybe a cause of batch effect. There you go.
Different sample preparation protocols.
Exactly, that's very important. So even if you're using completely the same buffers, right? When you prepare, if you prepare on different days, like your buffers are really a bit different, right? And even if you may not even realize that you're making small changes just because you had a coffee in the morning, right? Like this is why we are all striving to be a bioinformaticians, not fully wet lab biologists, but I respect if you're almost. All right. Anyone else?
Thanks.
Patience.
Patience.
But I don't think
Different technicians, you can see even this.
Exactly. Yeah. Just because you can see.
Kind of make your map with your iPad, you can see that, especially in time, yeah, or when you have the kind of pressure from biologists, so they say, okay, we have 300 people, but we have sequencing so far 200 of them, but let's do sequencing, let's do what's going on, and we'll do the rest of the further, yeah, forget about that. This will cause definitely batch effect, so definitely time again for that, and then delay depending on
Right.
the same.
Yeah, so you actually hit on 2 points that I want to talk about. One is like different people, but the other is like how actually you spread your like data collection points and like the data that you're processing. There you go. There we go. Okay. Okay, I think like we can slowly move on, but yes, like you hit some of the stuff that I want to talk about. Different mice, like the same strain but coming from different companies. Also, this is not a good experimental design if you have something like that. But sometimes you cannot help it because the data is not completely yours. Different sequencers. Would be great. Like for example, let's say you do a cancer study, right? So you compare your cancer against normal. In this setting, this is like what I'm familiar with, that's why my examples are like this story. You frequently would have a paired normal, right? So you would, during a surgery, you would get like a biopsy that is from the cancer and from the normal, right? And so you have paired normals and cancers. And you would like to see that a clear separation, right? Like this is even from the same sample. It's not like random normal samples, right? You would like to see a clear separation between cancer and normal. That's what we are studying, the differentiation based on biology. But frequently, what you would see is that the data like separates a different way, and instead of so like what I would like to say is like instead of jumping immediately to the cool differential expression analysis, it would be good to first take a direct look at the data. That's the best advice I have. And unsupervised learning is really good. So here is my favorite example of batch effect actually getting published. This is paper that came from Mouse and Code. Can you get a show of kind of people who actually worked on data coming from model organisms? Can you like raise your hand if you work on data from model organisms? Be that mouse, like Drosophila, whatever. Okay, who worked on mouse? Okay, great. So some people didn't raise for model organisms, but raised for mouse, which I guess it depends on your point of view. So this paper, published in 2014 from Mouse and Code, actually shows that all of your experiments are invalid because our assumption in using modeled organisms is that they actually are. We are able to draw conclusion about, for example, human based on the results that we get from mouse, right? But here, what they publish is in this PCA, the visualization isn't very good. I will show it a bit differently, but you can see clearly that dots, humans on left, are separating from mouse, and this is actual sentence from this is like one of the headers actually from the paper that shows that like gene expression is more similar for different types of human tissues, right? Then for the same type of tissue but from different organisms, which this just means that we should actually be studying human heart if we want to give conclusion about human brain and not, studying mouse brain, which obviously that's very sad conclusion. Obviously, a lot of people were not happy with this paper, even though it was published by very established authors, and some people later on did a proper reanalysis. And if you actually account for the batch effect here, like here on the left we have samples that cluster by actual organisms, and here right you have samples that cluster after batch correction. They cluster by tissue source. I don't know if you can see because this letters are a bit small. And what they discovered is actually that the runs that they made were not very well spread out. Like you can clearly see that the human samples were run together, right? These are different sequencing machines, different runs, different lanes here on the top. And there was only one lane where some of the samples were coming from mouse and from human. So that just brings me to the main point is that good experimental design is the clue to minimizing batch effect. And for this is it's very general, but that's basically what you need to do. It's you need to remember to keep track of the metadata, who is running the experiments, who is what machines. The perfect lab notebook, it's just priceless. And it's good that now we are getting them more and more digital, so it's easier. We need to be consistent about pre-processing of the samples, and we need to specifically take into account how the samples are. It's not possible frequently to pre-process all of the data. You have a big cohort of mouse. Extraction takes some time. You are not able to extract everything at once. You're gonna have small batches. But if you have them, try to balance the groups of interest. So to avoid the so-called perfect confounding. So, for example, keeping all of your treatment one in one batch, treatment two in the second batch, then you are not able to see what is actually a significant biological difference between the treatments and not coming from these technical differences. And if you can balance the groups of interest, try to keep at least one control at each time point, or each group, and then it's much easier to try to get rid of our batches. So how do we do it? I personally recommend Combat. That's a package from 2007. It was designed for microarray gene expression data, but it actually works pretty well for different some different omics as well. And in general, how it works is here you see normalization helps somewhat. You start by normalizing your data, but then if we color them by batches, you'll still see that there is a clear separation of batches. But after the batch is corrected, the batches should be more or less uniform, hopefully. And there is no better way to do it than unsupervised learning. You just visualize, then you cluster your data, you do dimensional reduction, you color your data by your batches. If you don't know what possible batches you can have, go through your metadata and try to color by all of the labels and see if you have this separation that you need to account for. Then run a correction and come back, visualize, and see if it's actually corrected. That's a very straightforward example, but as supervised learning, that's the way to go about it. And there is a lot of different packages. If you run Lima is a linear model, right? So it's not very complicated, but it can account actually for batches, including complicated batches. So if you have data coming from different centers, and inside of different centers, you have data preprocessed in different ways, for example, right? That's the nested batches. So that's the first example. And other than doing the dimensionality reduction, even straightforward looking at heat map, that's really like heat maps are your friends. This is if you actually plot the heat map of your data, even if it's like high dimensionality, and try to add labels based on batch, and this is like just clustering based on like hierarchical clustering, right? Sometimes you can see, sorry for the colors, this is from the original paper. You can see this separation. On left we have pre-batch correction and then post-batch correction. All right, so the next field where the unsupervised learning thrives is single cell. Yeah.
It was a loop on the pipeline.
Why there is an arch come back?
Yes.
It's because you should, like, you visualize, you see that you have a batch effect, right? You correct your batch effect, and then you visualize again to see if it actually was, like, if it's actually corrected, right?
But you identify again the correct and reverse over.
Yeah, don't go like over and over because you will never publish your paper, right? No, it's yeah, it's but it's very important to like come back and like not just apply the correction and be like, okay, I corrected it, right? Like just check if you actually corrected. If not, come back to the original data and try to see like maybe it's not an like maybe it's more complicated effect, right? Maybe more than one variable actually is impacting it. And testing for, first of all, for all of your metadata parameters to get a sense of what can cause it is the way to go. All right. So single cell gene expression analysis is the field that thrives on unsupervised learning. Obviously, like most of you are familiar with single cell experiments, and the whole idea is that like now with the advance in like microfluidics at the beginning, we were able to isolate just single cells, right? Or like hopefully single cells if we are talking about droplet-based methods. Like RNA exhalation and amplification to actually achieve like single cell resolution. And different cells, not only they look like different, they the idea, like the assumption is that they are actually different based on their transcriptional profiles. And this field is obviously moving very quickly. This is the first paper that I found that actually published a single cell method. It came out in 2009, and it published 8 cells. You know, this is Nature Methods, so you know it's not a bad publication. And it was a very good achievement. In 2016, 10x Genomics published the like brain cell data set that's over 1.3 million cells. So that's amazing. So how do you even look at data sets that has over 1 million data points, right? And 1 million data points / 60,000 transcripts for the human. So yeah, you apply unsupervised learning. You apply dimensionality reduction, you apply clustering, especially to identify different types of cells. And this is good because it's a data-driven approach, right? So we are not interested in just assigning pre-decided cell identities to our cells, right? We just want to explore and find the groups that are more similar to each other than to other cells. And this is like a very, I forgot to add the source. I will add it for you later when I give you guys the slides. But this is just very common workflow, right? So for single cells, you would start with some quality controls. You would like remove the cells that are like obviously are of lower quality and you cannot trust them. You would do some normalization, and there are different ways to do it. Like I talked to some people about this, and then most likely you would do some feature selection, and this is due to something that's called a curse of dimensionality. Did you hear about something like that? Can somebody explain to me what's the curse of dimensionality is? Curse. Yeah, it's a bit obvious and it's not that complicated, but this is like something that people like to say whenever they write single-cell papers, right? And this is because the problem is you have so many data points and so many features, usually, that with the amount of features, it's harder and harder to actually calculate meaningfully the similarities between cells, right? Like the distances, like the cells are getting closer and closer together the more features you have. Especially with the technical issues that are with single cell, which we're going to talk about in a moment. So frequently people would select just the top features based on variance. So you would remove uninformative features. Obviously, it always is going to include some of the bias, but that's the best way we found. And this is going to improve how our dimensionality reduction looks and how our clustering results are going to actually look like. So usually we would do dimensionality reduction at least one. Frequently we would do like PCA and then like TSNE or PCA and UMA to look at how your data looks like. And then yes, this is like similarities. Then you would usually use unsupervised clustering methods to find distinct groups of cells and then this is funny because you actually will assign them kind of like cell types, like cell identities. So, but it's not a supervised learning. Usually you use unsupervised learning and then you try to plot different markers of specific to different cell types and try to figure out what actually these clusters are. And actually, most of the packages, they use pretty straightforward methods for clustering, surprisingly, like k-means or hierarchical, so now you guys are already experts in this, right, after this workshop. And for example, if you use SC3 package, like one of the most commonly used packages for single-cell RNA analysis, you would use it. Actually, will run k-means for you using a Lloyd algorithm, but it's actually a greedy algorithm, so it will not only it will always find the number of clusters you ask for, regardless if their actions will be meaningfully different. It will if you run it multiple times, it will results will probably be a bit different. So what they do is like they run it multiple times and do a consensus of it to try to find like a more stable result. And it's important to actually check what your packages are running, because if you do, for example, k-means, then that means that the disadvantages of k-means are going to actually be, you have to take them into account. For example, k-means is biased towards clusters of similar size. So like rare clusters are going to be more, rare cell types are going to be more difficult to identify, and Casper is going to tell you something about this a bit later on. So here is like another cool figure that I like, that shows how unsupervised learning can be used for a bunch of different things in terms of single cell analysis. And yes, we mentioned discrete like cell type annotation and rare cell types. We can take a look at, for example, some specific gene signatures and plot them as a continuous variable and try to see how it changes for our cells. This is very important, like that our cells probably are going to be at different points of cell differentiation. So, for example, for hematopoiesis, we're going to see clusters actually getting, meaningful. The order of clusters is going to be meaningful, connected to our differentiation. And vacillation, this is like very beautiful word. So actually, like, if your cells are cycling, it's also going to have an effect on, and unsupervised clustering is going to help you with it. Just doing a PCA and of genes that are specific to a cell state in cell cycle is going to help you identify if you have cyclic cells and maybe you need to, as they say, regress out these variables, so like account for it. That's what goes into details, like account for it for your analysis as a source of a variation that you maybe are not studying. All right. So, like I mentioned, there is a bunch of actually, there's also spatial, but it's even more complicated, right? You add additional variable, like not only have a cell identity, you also have a space that the cell is coming from, for example, in a tissue, right? So that's just more and more dimensionality, right? And then I mentioned technical considerations. So I would like to ask you if you can tell me, especially if you had experience with single cell, what are technical considerations? What are the things that we need to think about when we are running a single cell experiment?
Running an experiment or analyzing data?
Analyzing data specifically, like because it comes from a technique, right?
I say like I mentioned the percentage of mitochondrial genes in cells. Pay attention to percentage of mitochondrial genes in cells.
Okay, yes, that's very good. I'm gonna give you one more candy. You want the other one? There you go. All right, yeah. Actually, if you have a problem with your protocol and your cells are dying, so right, and your quality, not only your data quality is going to be worse, like you're going to see the percentage of mitochondrial DNA. It comes from like apoptotic cells, right, getting higher, and this is one of the quality metrics that you need to take care of, like at the beginning of your analysis of single cell. True.
Hidden tablets, because if you do enzymatic digestion or any type of digestion, and you should do it consistently, but it's not possible because you use enzymes, it's biological stuff. So you need to consider how to get rid of tablets, because they are going to be a really big problem.
Yes, that's true, and tablets also go from because if you do like microfluids, right? You may not be able to capture just single cell. You will capture like two cells at the same rate, and suddenly you're all able to map the same cell in your data, right? The same identifier, and you will see that the expression is like double, but it's not going to be perfect, and there is actually a way, I don't know, like if you're familiar with single cell, you probably know that there's also another way to capture single cells, and this is by splitting and pooling, which means that by like probability, you like barcode all the cells, you pool them together, right? Then you split them again randomly, you barcode them again, and if you do it multiple times, I think 6 are enough for most of the datasets, the probability of a cell having actually the same combination of barcodes is so small that you are getting like single cell resolution. You can identify which data is coming from which cell, even though you just split them and pull them, and you don't have to do a microfluid extraction. True. I will give you a candy before I forget. There you go, a different one. All right. So what else? So we talked about enzymatic activity, right?
Well, that's one you already mentioned on your slide. The dropouts will have very sparse matrices, so like in one cell you won't capture like all the 60,000 transcripts that might be expressed in the cell, but you will just have a very small subset of them, and then you need to figure out is it due to like a deletion, so it actually isn't expressed, or is it just like a technical.
All right. Yeah. This is great. That was a hint. Yes, but also this just starts a topic, because you said that if it's the under expression, or if it comes from the actual technical difficulties. What are the technical difficulties? What is the where the dropout is coming from?
Sequencing depth.
Sequencing depth, yes. There you go. Something else? Like if you have a zero, so if you get your data from single cell and it's sports matrix full of zeros, well, what can be a reason for it to be a zero? Like other than like low depth of sequencing. Exactly. So in what way efficiency?
In like reverse transcription, PCR.
So amplification bias, yeah, exactly. So that's one of the reasons. I almost got it. All right. What else?
Maybe amplification power because of some mutations.
Because of mutation.
Some mutations.
Yes, actually, this is yes. This actually binds to what I wanted to say is that there is a cell type specific like capture efficiency, right? So like there's going to be some modifications for different cell types that are going to cause them to be less detected, like less captured than other ones. All right, let's go. There you go. This is workout for me and not for you guys. I don't know.
Length of genes. If the gene is long, it's easier to be captured than short genes.
Yes, exactly. So for like larger genes, like and higher expressed genes, it's more likely that you're going to actually see some expression. And for lowly expressed genes, it's more likely that it's not going to be enough for it to actually like register as an expression, right? So this is like some of this what we talked about, especially shallow sequence depth. But yeah, so these are technical considerations, and actually they also can be addressed with some unsupervised learning. And this is very cool, in my opinion, because like I said before, like this kernel density estimation is actually like I used it before just for like simulated data, but this is an example of how it can be used actually for for data analysis. So this is a package that was published in 2021 that already has like over 30 I think citations, and here they basically what they do is for some markers in general you're going to if they are lowly expressed genes, you're going to be it's going to be hard for you to actually identify this type of cells based on lowly expressed markers. So what they're doing here is they are using this kernel estimation. Sorry about math, it's not that important. What it does is actually it's like an average of, they look at the neighborhood, the nearest neighbors of the cell, and then they're doing an average. They're trying to like estimate based on like neighborhood of the cell what an actual expression should be there, right? And it's weighted. So this is like an example of how they analyze the CD4 expression, and they were able to find the CD4 T cells here and identify them. That's like a practical application of what is happening, right? So all of the dropouts actually impacts, like all the factors that we talked about actually impact the visualization in our dimensional reduction. But different methods can be used to actually work around this. All right. Another single cell example. I'm just going to give time for Kasper to talk about his cool research. If you want to, there is still some heavy.
For me, right?
For you.
Yes, as I promised yesterday, we were mostly talking about basic things, so there are a few more topics I'd like to sort of briefly discuss, but not go into too much depth. So, one of them relates to identifying rare clusters. So, in particular, we're looking at this in the context of cell types, so whether clusters would be potentially known or mostly unknown cell types, because usually what happens is there are a bunch of very well-known and prevalent cell types, but then if we're interested in discovery, then we might not be able to pre-specify all cell types a priori, and then we might want to use unsupervised clustering to detect something interesting. And why this is hard is, well, it is hard. And just to illustrate this, and it applies more broadly than just cell types to any kind of clustering is, so here's sort of a mostly synthetic example where there are 5 different cell types. And there are 2 that will become more rare than here they're at 5% prevalence, so these 2. And what it just shows is that, okay, here at least UMAP is able to separate them out. But if we now use exactly the same data, but we just sort of downsample the prevalence of these rare cell types to 1% or 0.5%, then what happens if we look at it here? If I asked you how many clusters there are, then it's not so clear anymore. Maybe you could say these are kind of a 4th cluster here, but you can't really distinguish between the 2 rare groups altogether. So this is kind of a generic pattern. And I would say that, in this piece of work, we kind of looked into how we can, in our specific scenario, how we can address this. But I would say that there's no sort of generic solution that you will always be able to find your cell types. So just to give you some insight into some of our findings. So you could ask, so here we use like a standard clustering algorithm that people use on single cell data called phenograph. But it can be any, like results will be very similar for any clustering algorithm. But the question is, if we run it once for a certain number of clusters, and now if we're unable to find rare clusters, what we can do is we can increase the number of clusters. So we can increase k and ask, oh, are there now more clusters? I mean, there will be more clusters, but will it be more sensitive? In a way, yes. But what we see happening is, so here are 2 clusterings. This has, it doesn't have an explicit k, but basically here there are less clusters, here there are more clusters. And if you look at how cells map in between these 2 clusterings, then it's just a mess. It's not like new clusters appear. What happens is this cluster got split into 3, but actually some clusters became together from different clusters. So it's a bit unstable, but also just increasing k isn't going to be a generic solution. So that's sort of one takeaway. And yeah, just to sort of reiterate that UMAP is not able to really necessarily tell us where rare groups lie. So this is a sort of example we looked at where we are looking at T cells. So this is a UMAP based on T cells only. And we can see that on this corner there are CD4 positive T cells, and in this part there are CD8 positive T cells. And in this corner there are Tregs. But now if we're interested in double negative T cells, so T cells that do not express neither one of these clusters and neither one of these markers, so this could be a potentially interesting cell type, then using something we sort of methodology we created, we can identify sort of 4 groups that are not distinct in UMAP at all, but then when we go to the sort of expression space and look at the markers, then they sort of do look distinct. But yeah, in general, whenever you're looking for rare clusters, it's a hard problem. Or in general, yeah, imbalance, a severe imbalance in the cluster prevalence. Okay, this was one point. Now, sort of switching topics, talking about something completely unrelated. Something we talked about yesterday is how to choose K. Clustering method is an everlasting question, no generic answer. Just to give you an idea that there actually exists a research field that kind of tries to circumvent this question. The area is called Bayesian non-parametrics. And if you have heard of Bayesian statistics, it's kind of a sort of separate take on creating models where we capture uncertainty using distributions, uncertainty about parameters, etc. So there is a way to construct models where we say that we don't exactly know k, but we allow it some uncertainty about this parameter, or we allow it to be potentially infinite. And actually, you can make this work. It will involve some maths to understand it, but actually, it's not that hard. Like, there are ways to make these things work quite nicely. So, for example, Bayesian non-parametric models have a property that you cluster your data, but now new data comes in. What should be allowed to happen is it doesn't necessarily have to map to all existing clusters. Potentially, a new cluster is allowed to arise. So this is what kind of this infinite number of clusters allows. And another thing we might want to say, oh, actually, it's not necessarily a fixed k, but we can say with 80% probability k is 5, but it's also plausible that it's maybe 6 or maybe 7. So I will not show any maths, but if you're interested in this thing, you can read up. There are some, I can point you to some tutorials. But yeah, broadly, there are sort of infinite processes in this field. For example, something called the Chinese restaurant process, which is a way to set up a generative model for the number of clusters. And the way it works, this is a gift from Wikipedia, is that I'm not sure if... Oh, it's playing. Okay. So basically what happens is we start generating clusters from scratch. Let me start this again. Start generating clusters from scratch, as in there will be tables in the Chinese restaurant, and first the customers can either join an existing table or join a new one. And this GIF is playing in a weird way, but yes, so here, first cluster, second cluster, third cluster, fourth cluster, etc. So this is a generative process, kind of, which can result in a different number of clusters. It's not sort of pre-specified by us, but it allows a flexible number of clusters. So just that you have heard of the fact that there exist ways to kind of circumvent these questions, but again, there will be some assumptions somewhere, so it will probably not be like a universal solution to avoiding any subjectivity.
Is this model specific?
Sorry?
Is this specific to like a certain type of models, or can you use it for every method?
So usually you would see this being used, say, for a Gaussian mixture model, which is kind of like an extension of k-means. So there you probably find even an R package that implements that, so you can just run it. If you want a different model, then it might involve some more work. Okay, and third thing I wanted to talk about, again, different topic now. So yesterday we talked about PCA. So just to tell you that there exist sort of two types. We can extend PCA in two directions. One is to make it probabilistic, or Bayesian, and the other is to make it nonlinear. So briefly talking about both sides. So the way we think of PCA usually is it's something that takes in the data and it maps it to a low dimensional representation. So in this case, these 5 images get mapped to these 5 locations. The way usually probabilistic models are specified in the other direction. So they start from saying, it's like the generative perspective, which says there's a low dimensional representation, say something about how tilted the digit is specified in the low dimensional space, and there is some uncertainty about it, representing the distributions, and then it ends up being mapped to the data space. Even though we know that we can also reconstruct with PCA, so it's not that big of a difference. But with this class of models, we can do some cool things. So we have the generative perspective, which is just the interpretation. and we can have some uncertainty in the model and be useful. What's nice is, actually didn't talk about it much, but PCA doesn't really work when you have missing data. So you have to do something about it. With this, so the probabilistic extensions, which originally come from this paper from 1999. You can have missing data without doing any imputation, so that's nice. You, again, choosing K can be more flexible, as in you can basically avoid choosing the exact K value. And also, if you have binary data, count data, other types of data, then continuous, then there are nice extensions. So it's more like a sort of building block how you can set up various models. For example, there's like a popular multi-omics modeling framework called MOFA that kind of extends probabilistic PCA to a multi-omics setting, etc. Okay. So this was for probabilistic extensions, and now about nonlinear extensions, just also very high level description. You haven't had much deep learning, there will be more towards the end. I don't know if there will be autoencoders there, but basically what an autoencoder is sort of this pipeline that takes in your data, then maps it to a low dimension using some neural network, and then it reconstructs it from this low dimension back to the data. So actually PCA is not that different. So it turns out that if this thing, mapping your data to low dimension, is linear, and the other thing is also linear, then you end up something that is very similar to PCA, with the only difference that for PCA, PC1 is like the direction with the largest variance, but here you actually don't know where PC1 will be. So up to like a rotation, it will be the same. So now you can imagine that if you allow nonlinear mappings, then you end up with a nonlinear extension of PCA. And there's a lot of work in deep learning doing this on various types of data and various extensions as well. So for example, going back to this probabilistic model that ends up with this direction mapping, it will also involve mapping from data back to the low dimensional space. Again, yes, if you do these mappings with neural networks, then you end up with autoencoders or variational autoencoders. But variational just means that it's like the probabilistic version where you have uncertainty as well. Okay. And these kind of models have also been used for single cell genomics. So there are some papers. There's a repository with a toolbox. So this is probably one of the first papers for this sort of field. I will not go into detail. Basically, it involves setting up the model architecture, and actually it's a little bit more complex because they have count data and they want to model both the dropout as well as like the library size. So therefore they have a bunch of neural networks, but the overall idea is basically an autoencoder model. And they also have the functionality to simultaneously regress out the batch effects. So batch effect, as in if you have your batch labels, then there's functionality to take it as an input, and therefore they can show that when you take their low-dimensional representations, this is one of the examples from the paper, and you color it by batch, then you can see that there's no more batch effect anymore because they were able to deal with it. But broadly, there's like a toolbox called SCVI tools, which has a bunch of implementations. I don't think they're particularly easy to use, as in, it's not an R function, it's more like a Python, I guess it's a library, but I haven't used it much myself. But if you're interested in this, then it's a pointer for you here. And that's all I wanted to say right now about these topics. Yes, so I'll.
Awesome. Let
Thank
me give
you.
you some candy to go.
Oh,
Thank
you.
All right.
Yes.
Imagine the problem which also increases in distance, that we have much more cells, so which means much more samples, but less features, actually. Or imagine flow cytometry, where you can reach even millions of samples that block amount of feature, and what is your experience? Just your experience, which would advise as a custom method. For some cases where you don't have much features, or with this kind of proportion.
So you mean like to have cell surface markers, right? And like some small amount of features, and you still want to like classify yourself.
Isn't that a typical ML problem when you have way more samples than less?
So something I wanted to say was actually these kind of autoencoder tools for single-cell genomics. One reason they one selling point is that they scale very well to large number of cells, and that's because, as is common with these kind of models, you can usually train them using subsampling. So you don't necessarily even have to have your data in RAM at once. You kind of, because at every step you kind of take a subsample and you update your model. So they scale very well for that. But regarding clustering, I don't know if there are what other bottlenecks.
Interesting. I would say like the more classical state would be if you have like gene expression, so you have a bunch of features, but you have additional like some markers that you are using for that like drive your cluster separation, right? But that's in my experience.
Well, I think it's harder actually. It was harder to work with the data where you have way more features than you have samples, right? Because that's the thing that we have to struggle with initially. So if you're going to have it the other way around, I don't see any problem with that.
Yeah.
In principle.
I guess it's more like a runtime problem you face. Or
Yeah, we can talk about this a bit more like during coffee break or something.
Yeah.
Try to, all right. Yeah, we have significant amount of time, so I should be able to show you not only method but also some biology, which I know some of you probably missed. So this is my last example. That's practical application, and this is my own research for multi-omic data integration. So, like I mentioned before, the introduction, we want to integrate different omics to get some more insight into like systemic functions of an organism, and usually when we talk about multi-omic data integration for the purpose of molecular subtyping, what we are talking about is we are trying to build a low-rank representation of integrative representation of your different omic data sets of your samples in these different scales, and so this is a dimensionality reduction, pretty obvious because we are looking for a low-rank representation, and then we are doing a clustering of sorts because we are looking for groups of patients that are more similar to each other than to different patients. Because finding groups based on this integrative representation can actually be a hint of, analyzing the difference between them can actually be a hint about possible biomarkers that could be, or like driver genes that could be used to investigate and like develop new therapies for disease, right? So the first thing that we need to talk about when we are talking about multi-omic data integration is the fact that, well, it is challenging, and there is a lot of things that could go wrong. So I would like to once again open the floor to you and ask you, what do you think are the challenges of multi-omic data integration, of trying to find this like integrated representation of different types of omic data, like methylation, expression, chromatin accessibility, mRNA expression, what have you.
Missing data.
Who said that? Okay, missing data. What do you mean when you say missing data?
That usually to get new mixed data, you need to perform a lot of experiments, and in my data set, not for all individuals, all kinds of experiments were performed. So actually, there's like a little sample of people who actually have only experiments.
Okay, so what we're talking about here is it's what I would actually call incomplete data sets, right? Which means that there is just a small overlap, right? Like you don't have the same omic data for all of the samples, but you have patients for that you only have like one data type for, or multiple but not all. And this is actually visualized here by this node, just didn't have any connections to other ones in this one layer of data, and that's correct. That's definitely one of the very big challenges, and a lot of methods actually. Oh, nice. Do not account for this. They require you to have a perfect overlap, right? And if you look at the data that's available publicly, so the perfect example would be, for example, the cancer genome atlas datasets, right? You will see that the data was pre-processed over the years in different centers, and sometimes there was just simply there wasn't enough material coming from a patient to get all of the data types. Yes. What else? Batch effect for sure. There you go. Easy one, but I respect. Are you paying attention to the slides? Something else?
The other way around on Java sets, so not all the features are measures because of arms corresponding.
Yeah, so like simply just missing like NAs in your gene expression, right? Like missing values that either like if you do single cell, there was a dropout, like that's clearly missing value, or is it missing value? Maybe it's under expression, you're not sure, right? For bulk data, which is what I was focusing on in this project, you will still have some missing data, and it's actually surprisingly a very big issue. Like there's really a small amount of multi-omic data integration frameworks that support missing data from feature matrices, and most of them actually require an imputation. And every time you do an imputation, you need to remember that there is a potential to include bias, right? Here we go. I don't know, it varies. Okay, what else?
Mixed subtypes, for example, we can classify patients into different subtypes at the same time.
Mixed subtypes. Okay, so in what way?
Well, for instance, in colorectal cancer, you have 4 subtypes, and the mixed subtype would be patient who they have the features of the subtype 3 and 4 at the same time.
So I would call it more like, so obviously we are talking about clustering here, so these are the distinct groups, but the biology, it's not always so easy, right? So that is more like a continuous space of where the patient actually is classified. And this is because of our assumptions of biology, first of all, but there's also, there are some cancers that have more distinct subtypes only because there are actual treatments that patients are going through that impact the differentiation between the samples, right? And yes, the mixed subtypes technically correct, I will give you a candy, but technically it's not a challenge of the integration, like finding a representation. It's more of a like ambiguous sample getting clustered, and you're not sure in which cluster it is, right? Yeah. Okay. There you go.
Different scales. because, like, for example, changes in expression level, we do not know how it corresponds to changes in iteration level and in chromatin accessibility level. And of course, we will scale our values, but then we will miss this. And actually, we don't know how the changes in expression value will affect changes in other omics values.
Okay, so there are two things that you touched here. The first one is the fact that like the scale of the data is different, right? So let's say you're running an like methylation, like Illumina, like methylation experiment, human methylation 400K. You have 400,000 features, right? 400,000 probes. Not only I almost gave up the problem, another problem. It's and then you have like expression, let's say, right? An expression if you actually look not at a transcript but at genes, so protein coding genes in human, that's like around 20,000, right? Features. So how are you actually like going between this? Like how are you integrate data set of such different scales? Yes, that's true. The other thing that you actually touched on is that our different omics, not only they show different snapshots, they are also frequently moved in time, right? So you have like a methylation, and then the methylation impacts your expression, like later on. It's actually so the moment that you actually get a sample may already show the change in expression, may not show a change in expression yet, right? And this is a design like challenge of multi-omic data integration, but it's also what we are actually studying, right? So I always give up one, maybe.
I guess it's also a problem that we are not analyzing exactly the same bit of tissue because we usually can't. Maybe it's getting possible, but for example, if you have a FFPE, for example, and to slice it, you have to get like different levels. It's not exactly the same one, so it maybe do some
It's very nice. I don't have it on slides, but you're correct, because it's actually in multi-omic data integration we sometimes talk about multi-omic versus multi-view, and like there's more and more platforms that actually allow you to profile different omics at the same time. So for example, from 10x you have a multi-omic that allows you to profile expression and accessibility in the same cells, right? So you have the same omic, but frequently you're correct. Like there's going to be slight differences, but we try to assume that at least like in bulk, that the differences between individuals will still be bigger than differences in like tissues coming from this technical issue, right? But yes, that's a very good point.
I don't know if I'm going to say something stupid.
Go for it.
I forgot the word in my mind, but there's some kind of splicing, like when you have different genes and they not necessarily certain expression of certain genes has to be connected to certain proteins, so maybe that's also hard when you measure specific genes and you think that's connected to this protein, but it's not.
I think it's connected to what, you're correct. It's not what I was getting at, but you are still correct that if we are looking at different omics, like different aspects, right? And if we don't have a full knowledge about like what's happening, frequently we need to remember that this is still unsupervised learning, what we are talking about here. So we are looking for those exact patterns, right? That's what we are trying to discover. All right, so I kind of hinted at the fact that like different sizes of features, right, in different omics. And if you have, let's say if you run ATAC-seq would be a good example, then how your data looks like. Well, the data is going to be predominantly certain value, right? It's going to be predominantly 0. So how do we actually call the data sets that are predominantly zeros? Sparse, yes. A lot of you, sorry, no candy. Yes, sparse data sets, and if you do like a taxic experiment, for example, most of the regions that you are actually looking at are going to be like zeros, right? You don't have counts there, so your data is sparse. The good example of this is also somatic mutations, right? Somatic mutations is binary data. Most of them you're going to have like unmutated, and then for some you're going to have like one, right? If it's mutated, for example. And how do you actually integrate this type of data with continuous data, like expression? That's another problem. And the last problem I wanted to touch on is the feature selection, right? Like again, if we have omics of different types, if we have 400 probes for methylation and a certain significant amount of probes is like slightly variable, are you getting rid of them? Are you doing additional feature selection? This includes some bias, but no, not all of the models are going to actually be able to like support this type of data, right? Like actually, I think I had problems opening like this type of methylation data in R, just opening it, right? Because it actually like opening it classically takes twice the amount of memory that it normally took to work on it. So R. All right, so missing values in incomplete data, feature selection, sparse data sets, data heterogeneity, that's like a topic that's, well, it encompasses all of the like biological considerations and technical ones. And the fact that because we have data that has completely different distributions, we probably need to like normalize it, and there's different normalization techniques for that are omics specific, and how do we actually work with this? So there are different approaches to do to creating like integrative representation of multi-omic data, from very easy ones where you just concatenate different omics, right, and then try to cluster them together. So obvious problem already, like pre-processing, right? How do we actually, and if you have data of different amount of features, like how do you, like one data is going to completely take over your data, right? Like one omic is going to completely take over your data. Then these are so-called early integration. Late integration would be you try to cluster separately, and then you attempt to find like consensus clustering, which is very complicated. There are different ways to do it. Then we have similarity-based methods, which means that you investigate like omics separately, try to find the similarities between samples, and then you try to integrate these similarities. Well, there is one package that is very like famous, which is called SNF, Similarity Network Fusion, which would just take those similarities between samples and then try to like fuse the two different networks together and drive conclusions based on that. Then we have all of the dimensionality reduction methods, similar to PCA, but even different models that we haven't even talked about yet. And then probabilistic modeling, which seems like it encompasses the most of the heterogeneity and complexity of our task. But the problem is, usually you have to assume certain distributions of the data. So sometimes it's like limited to certain types of data that you can actually analyze. So that's the background. And the method that I developed and the novel framework for integration, it's called SUMO. It's subtyping using multi-omic data, and it's available at GitHub and on like PyPy as a command line tool. And it actually connects two types, like similarity-based methods and dimensionality reduction. All right, so the whole framework works in 3 steps. Actually, the main method is here, and this is just a downstream analysis example. So what we do is like first we investigate all different omics separately, and we calculate pairwise sample similarities based just on certain data type that we have. And at this point, we already have we don't have feature matrices anymore, so we don't really care about our different omics having different number of features, for example, or different pre-processing, because we are working just using similarity matrix, right? So we have sample per sample here, and this can be also like shown using multiplex graphs, but just like we can talk about this during a break if you want to. But just thinking about the similarity matrices is enough for now. Then we actually do like a factorization using this is non-negative matrix factorization, which is a different type of dimensionality reduction, and I'm going to show you, like, explain it in a moment, and use a consensus clustering approach because, again, it's a greedy one, right? Like, it's not fully deterministic, so we want to run clustering, basically, dimensionality reduction clustering multiple times, and look for different starting points and try to find a consensus, a stable result. And then we can do a dimensionality, like the downstream analysis. So the intuition here is that we already pre-process the data separately, so we don't have to we sidestep a bunch of issues, right? And if two groups of samples are going to be different from each other in similarity matrix, if we take a look at it, it's going to have like visible communities, right? And if the data is more noisy, based on this omic, it's going to look like less decisive here, but it can still give us an insight in combination with different layer, right? And it all comes down to the lower rank representation. So what Sumo uses is actually non-negative matrix factorization. In matrix factorization, what we do is actually we are looking for different matrices that when they are multiplied by each other, they are as close to our input matrix as possible, right? So this is a straightforward example of an optimization problem, right? We are trying to optimize the difference between the left side and the right side. And actually, this type of dimensionality reduction is very popular in this type of optimization problems because we very clearly we can try to impose additional constraints on our matrices. For example, non-negativity, as here is shown, and for example, we can try to make one of these matrices low-rank matrix, like we can constrain it, and make it as sparse as possible, and this gives us a low-rank representation of our data. Classically, this was done using feature matrices. What we do is we use similarity matrices. And basically for every optimization problem, what we are trying to do is trying to find a global minimum instead of a local minimum of our function. This is a 1D example. It's very easy to, if you've never done any optimization problem before, it's very easy to imagine like a blanket on the table, right? If I just throw a blanket over this, we're going to have like a valleys and a hills, right? And what we are trying to do is try to find this global minimum somewhere of our function. And this is done usually by like differential equations, right? But the intuition would be that if you just start by random values, the difference between your right side and the left side is going to be huge. But then we can try assess how big the difference is, make some small change in our matrices, assess the difference again, and see if we're moving in the right direction, right? That's what we are talking about here. And Sumo actually uses a bit like modified version of this, and what it does is that it shares this one H matrix is shared between different layers, which means that this is our integrated representation. It's shared between layers, and this S matrix is omic specific. It just tells more about like relationships between clusters. Again, Kasper mentioned the selection of K. K, or like number of clusters, is called in factorization clustering rank. And here in this example, we are doing this very greedily. We're going to cluster using different amounts of clusters and then see which of the solutions is the most stable. And this is the solution that we're going to select. All right. So we are doing this factorization. What we are actually doing a couple of more neat tricks, if I can say so myself. And this is actually the consensus clustering. So we're going to run this clustering multiple times, starting from different like starting points, like semi-randomized initialization actually, and then each run is going to actually be run on a subset of our data, while we are making sure to like cluster every sample at least a couple of times. And at the end of each factorization run, we just get the representation of the samples in our lower-end space, which we can cluster, and we can get cluster labels of our samples. Then the collection of cluster labels can be used to create a consensus matrix, which is just created by checking how many times different pairs of samples actually end up together in a cluster. Right? And we can even create multiple consensus matrices just by taking different subsets of our runs, right? This is how we find this cool error bar. I know, I'm getting excited about different things than you guys probably. All right. So we cluster our samples, we have our integrated representation, and we have our groups. And then we can actually do some downstream analysis. If we have our groups and if we have our initial feature matrices, what we implement here is actually a gradient boosting classifier that takes our feature matrices and try to find the features that are going to support our cluster separation the best. And that's an example of a downstream analysis. It actually comes from a different paper. This is our protocol that just shows how to use our package. All right. Before we go to the rest of the presentation, any questions?
So actually I have two questions. First one is, do you have any limitations on the number of individuals that actually should be studied? Because I know that some integration methods are designed for single cells only, and the authors actually don't recommend using it for bulk analyzing the small samples.
So this is opposite. Like this was designed for bulk, which means that you can still use it for single cell. It's just gonna take a lot of time. The problem is that we are running this factorization multiple times, right? So that's the limiting factor for sure. And then but you're going to get results at the end, right? The other problem would be that if you are doing single cell data, calculating a similarity matrix will be a bit more complicated, right? Because you have to calculate it like this is going to be a very large matrix. So you're going to need more resources on your machine. But also here, so the whole idea is as long as you figure out for whatever data that you're using, like you can use something completely different, how to actually calculate the similarity matrix, you can use this package. Like the rest is like universal. The problem is how to actually calculate the similarities. And here I'm going to show an example, which is a bulk count, normalized count data, so like gene expression, methylation, and mRNA expression, but for which we use Euclidean distance. But we actually we implement a couple of different similarity metrics, and for example, if you do single cell ataxic, like cosine similarity, for example, would be better than Euclidean similarities, right? Yeah.
So the second question was, is it implemented in R also or not?
That's Python package, yes. It is a Python package, but it's a command line tool, right? So you can run it from command line, and it creates, at the end, you get all of your NumPy arrays that you can technically read in R, using reticulate, and that's what people are going to do at my hackathon. Spoiler. All right.
Have you used it with proteomics data as well?
I haven't yet. I'm super interested in it. I think, yeah, we should talk. I think using it would be possible with quantitative proteomic data, coming from mass spec, for example. However, the problem is the level of noise is completely different then. So it comes down to calculating similarities in an efficient way. So, yeah. All right, we have a couple more minutes, so I'm just gonna run quickly. I wanted to give you more candy, but sad. All right. So the whole thing about this part of the presentation is this is a new machine learning approach, right? It combines different unsupervised learning methods, but it's a new machine learning model that we created. So how do we actually you learn about cross validation and how to test your models, etc. But how do you actually go about creating your new model, right? And the thing is that the most important lesson is that you shouldn't underestimate the value of your of simulated data, right? If you can try to create a toy example of what you are doing at the beginning, and you know the grand truth of it, you should just test it using this first. Test your ideas. It doesn't cost much. That's the first thing you should do. Test if your idea has a potential. Maybe this data set is not going to actually show you a full scope of a problem, but at least you will see if it has a potential. And so this is the first thing. Second thing is how to test it. Consult your goals. Our goal was actually to address the limitations in for example, imputation, right? Our model does not require any imputation and any feature selection. You can still use it, right? So we actually, when we wanted to test it, we would test if the performance using the data with increasing amount of missing data, for example, and to see how actually robust it is to missing values. And then, yes, obviously, compare with competition. That's very good. Find the best models that they are. Do not compare with the models that are shitty, possibly, because that's what we all want to do, and that's what people tend to do when they publish, just to show in the best possible way that your model works. But yes, compare with competition. Know what are the limitations of your package. Repeat each test many times. Error bars are very important, especially if you have, in this case, the package is not deterministic, right? So it actually the results can change slightly, but this is why it's important. If we have to check if our results are stable enough for it to be trusted. And technical consideration, we talked about this. Specifically, I wanted to mention that in this last part, in the downstream analysis, we actually need to train on a whole data set, so it's going to take a lot of memory and a lot of time to actually do the downstream analysis, which is outside of the package, but it's still important. All right. So that's the example, right? So our considerations were incomplete data and data that's very noisy. And so this is why we compared with different packages, so this is a simulated data set. Each dot is actually 100 repetitions for each of these packages. It took some time. But we tried, and this is a median of a metric that compares our cluster for samples and clusters that are coming from the grand truth because it's simulated data. And you can see that some of the packages, for example, this is a cluster bias, which is a gold standard for many years for multi-omic data integration. You can actually see that it makes certain assumptions about the data, right? So it starts from the worst results from the get-go, even though the noise is not so high. And here we are checking for the overlap between different omics, and up to a certain point, it performs pretty well. Then this is actual. I'm not going to stop on this, but we actually compared using different biological data sets and tried to see if we are able to find subtypes that have different survival for different types of cancer in TCGA and different differential significant enrichment of clinical labels. And it performed pretty well. And here I promised you some biology. I'm out of time, right? So just a slight mention of and just a real example of how it looks like. So this is a downstream analysis, and using our method using TCGA low-grade glioma dataset. That is expression mRNA, expression DNA methylation using the total of 556 primary tumors. So here's a quick information about lower-grade gliomas. They consist of around 5% of all primary brain tumors, and when you present with a lower-grade glioma, the symptoms usually include like seizures and cognitive impairment, especially if you have a glioma of a higher grade. And using the WHO guidelines, usually the diagnosis is done based on histology first. So they look at certain features like the necrosis of cells, for example, as a hallmark of glioblastoma. And then in 2016, they actually did a study that suggested to include different like specific mutations and markers. And this is some of them. I would like to highlight this one, the 1p19q codeletion that actually like differentiates here the gliomas of grade 2 and 3. They sometimes, like the differences between them are not so high. Alright, so when we run sumo on this is how we actually select the number of clusters. Like I said before, we do it briefly. We run for different ranks, and then we check how stable the results are. What we are using here this is a cognitive correlation coefficient. It's a metric that's specific to the non-negative matrix factorization, and we want it to be high because this is the like Pearson correlation between sample specification and their hierarchical clustering. It's a bit weird, but it's like specific to NMF. So we actually took advantage of the fact that we are doing consensus clustering, and we defined our own metric, which is proportion of ambiguous clustering. So essentially what we are checking is for all of the pairs of samples, what are the samples that sometimes are together in the cluster and sometimes they aren't, because we want them to be a small amount of them as possible. So we minimize this. And we find two possible solutions for 2 and 5 clusters. And for 2 clusters, it actually recapitulates pretty well the separations based on biology that WHO uses. And this cluster that has the better prognosis, it includes the gliomas that have the codeletion that I mentioned, right, and the IDH mutants, or they don't have codeletion, but they have a hallmark of like higher global levels of DNA methylation. And here for 5, this was very interesting for us because we saw this one group of patients that were separating pretty stably that had different survival. So it all comes down after this integration to actually taking a look at our groups and trying to find what differentiates them and if it's actually, a biological difference. And what we find is actually, even though this patients would mostly be classified as grade 2-3, so they wouldn't be given as much of an aggressive treatment. Some of them that were separating, their prognosis was much worse, and this is because they had some of the hallmarks that are very similar to like glioblastomas, much more aggressive tumors, and this all comes down to actually this modification, which is chromosome 7 gain, chromosome loss, and on chromosome 7 there's actually this is one of the examples. There's a gene that's epidermal growth factor receptor, which is the point mutations and amplification of these genes, this oncogene, are very common in glioblastomas. Like around 60% of glioblastomas have them, but they are rarely uncommon in lower grade gliomas. And on chromosome 10, there's actually P10, which is like the deletion. So if we have a loss of chromosome 10, there's going to be a deletion of this gene, and this gene is a known tumor suppressor, which may also explain why these patients have worse survival. And the other hallmark that we saw was actually like lower methylation overall, which is also connected to glioblastomas. I'm going to skip most of this. What I wanted to say is that we did some additional additional analysis because this is a bulk data and tumors are a milieu of different types of cells. So what we wanted to hypothesize here is that environment plays a role in creation of this different subtype. And when we actually took a look at the immune cells and stromal cells that are in these different CGA samples, these are X cells. This is X cells data. It's based on gene expression. Then our subtype 2, which is the differentiating subtype, is much more similar to glioblastomas than to other lower grade gliomas. And here, this is the methylation analysis, so you can see that it's much different. And this is like some other subtypes. I'm happy to talk about this with you separately. So, conclusions. The most important advice I got was that heat maps are your friend. Whatever you're doing, even if you're doing like higher complexity data, take a look at your data. Either just do a heat map and like plot all of your variables that you're checking, so for the batch effects. Just a small, even if your data is complex, just hierarchical clustering sometimes can reveal a lot of insights. If you can do like PCA, try to see if there's a separation based on batches. Keep track of your metadata. It's very important for batch correction, but also in a perfect world, you as a person that analyzes data is going to be a part of a whole making decision of how experiments are going to be made. And we all should strive for this, but we know how the reality is. And if both you and an experimental person are keeping track of the metadata, I think it's going to be much easier for you. So analysis or experimental design, that's essential. Next, each model has assumptions, so whether you are developing your own method or you are using different methods, models have assumptions and you should check for them. You should make sure that you're not using in one after the other methods that have different assumptions, and then at the end you cannot really trust the results that you're getting because you're not sure what would happen during the analysis. And don't underestimate the value of simulated data because this is how you check your assumptions, not only the model assumptions. And the last, possibly the most important conclusion from this talk is, if you're interested in multi-omic data integration, come to my hackathon. And thank you. Acknowledgements, especially for these people. They are mathematicians and they actually checked all of our formulas. It's very hard to develop new models. And this is my current affiliation, and some of the funding came from these institutions. Questions? No questions. All right, dinner then.